So as Pablo mentioned, in 2015, we were working on the original Dream Olfaction Challenge. And this was, I'm super happy to be back here at the Dream Challenge. That not only, I think, crystallized the field around this problem, but also introduced me to a lot of collaborators like Pablo, Rick Gerken, et cetera, people who competed in the challenge and worked together to solve these problems. So it had a really big impact on me, and I'm really happy to be back. So the key problem I'm going to be talking about is sort of grew out of this challenge and was part of that challenge is how do we look at a molecular structure and predict what it's going to smell like. I'm going to be talking primarily about single molecules. In this talk, the challenge was primarily about mixtures. And the sense of smell is really important for both for survival and for social interaction for the vast majority of species on the Earth. And for humans, it's really critical for overall health and for quality of life. So you can think about a situation where we eat a meal 3 times a day. That's important not only for nutrition, but also for connecting socially with other people. Is the sound terrible?
Maybe if we turn off the amplification, I don't know where it's going to work. I think it's the acoustic control.
I can do that. I can lower it here. Okay. Go ahead. Oh, sorry.
All right. So despite the importance of olfaction for our, yeah, better. Despite the importance of olfaction for quality of life, it's really pretty poorly understood relative to other senses. So if we look at digitizing images, we can type this sentence in, Alexander Graham Bell discussing odors with Hermann von Helmholtz using a smellophone. And of course, we can now use an AI generator to create this fictive scene. So in Vision, we have this incredible control of, we've digitized images, we know how to translate text to images, and meanwhile in olfaction, the situation is pretty different. So Rick Gerken and I have been part of a project called Perfume, which was to organize a lot of different data sets where people have described odors. So in this case, I'm going to look at 7 different data sets. These are authoritative reference sets of what something smells like for the odor of 1-octanol. So the Leffingwell data set, this is a person deeply in the flavor and fragrance world, published a product that would describe an odor. He describes 1-octanol as orange, rose, sweet, and waxy. Good Sense is a website that was a lot of people in the flavor and fragrance industry use as a reference. Here's the description from that. You can see it differs from the original. Here's AromaDB, which just describes it as citrus. The IFRA describes it as green, fresh, and floral. Arctander is a famous perfumer who smelled a lot of molecules and wrote observations down. And this is the Sigma Flavor and Fragrance Catalog if you want to buy flavor fragrance molecules. And this is a Dravnik's Atlas that has a quantitative rating with all these. And so has a number after each to tell you how much that applies to it. And you can see that this is the same molecule. So in Vision, we can go create an image turn our text and image. In olfaction, we can't even really describe what something smells like in a veridical way. So why is that? In Vision Audition, we know what the axis, relevant axis is. We know that wavelength is relevant to color. We know that frequency is relative to pitch. We know that there are limits on these, right? So we know that the visible light spectrum ranges between 400 and 700 nanometers. We know that the audible spectrum is between 20 and 20,000 hertz. In addition, we know how to mix these elemental stimuli together to make other stimuli. So I can mix red, blue, and green together to make some yellow color by changing the proportions. And in Audition, I can mix elemental pitches to make chords, major chords, and minor chords. And we understand how that will be perceived. In addition, in Vision Audition, we have tools that measure physical properties and report the perceptual properties. So we have a colorimeter that we can point at something, it will tell us what color something is. We have a spectrum analyzer for Audition. And in olfaction, sort of the ground truth that we use, there's no tool that will do this. Instead, we rely on a human panel. So the human panel will smell this and describe what it is. So though we have some analytical chemistry tools that tell us what is there, we don't have any tool that can yet translate to perception beyond a trained panel. So why are we using sensory panels instead of some sort of instrument? It's because we don't really understand the mapping between chemical structure and perception. So I think that's really vivid in these depictions of, from Charles Sell. He made these triplets. So here you can see there are two molecules that are similar in structure, and one is the oddball out in terms of chemical structure. And here you can see that these two are more similar to each other in perception, and this is the oddball out. So why is it that we have this mismatch where things that look the same can smell different and things that smell the same can look different structurally? Here's another triplet. So here are two molecules. These have exactly the same atoms. They're just rearranged in different ways. This is a straight chain. This has a branch in it. And this molecule is quite different. It has 6 chlorines on it. And yet the two on the right smell like camphor and the one on the left smells fruity. So in this same paper where Charles Sell talked about these triplets called On the Unpredictability of Odor, not only did he make the case that it was very difficult currently to predict what something would smell like, but he also made the case that our ability to predict odor properties of molecules will not improve significantly in the near future, right? So here I am telling you about how we're running data challenges on this. In 2006, Charles Sell argued that this was going to be very difficult. So why did he say it was difficult? Partly because at the time, the human olfactory system relies on 350 different types of olfactory receptors to sense something. And we were very poor at predicting how any one molecule will bind to any one receptor. So now if you're going to combine all 350 receptors with at least thousands of commonly encountered odors and try to understand all those mixtures, it would be very difficult. But of course, at the time, right in 2006, there was not the sort of preponderance of machine learning to attack problems like this. So here I'm showing you a graph with a year on the y-axis, and it's showing you 4 different breakthroughs in machine learning technology. So Google Translations, the ability to be a human at Jeopardy or video games, and the ability to identify objects in an image. And you can see that in all these cases, the algorithms were developed before 1995, and the breakthrough didn't happen until people collected a really large data set. So once you had enough data, the progress toward a breakthrough happened very rapidly. And so we think that olfaction is not a more difficult problem than these 4 problems here that people have had huge breakthroughs in. But in fact, we think that the main problem is that we just don't have enough data in olfaction. So I'm going to talk a little bit about this. I frame this as digitizing odors. How do we take an odor and represent it numerically, its perception? I'm going to talk about predicting if a molecule even has an odor. This is a problem that was largely unsolved for many years. I'm going to talk about variability in odor perception from person to person. And I'm going to talk about work predicting the odor of a single molecule from chemical structure. So first, we're just going to talk about whether a molecule has an odor at all. So just going back to this visual analogy, if we talk about the visual spectrum, a photon of light can have many different wavelengths, and only a small band here is something that we can perceive as color. And so when we understand that, that allows us to draw a border around the stimuli that we have and ignore everything outside of that border. And it allows us to evenly sample within that space. So now we know exactly what that space looks like, and we can set our wavelengths up. So if we're looking at a neuron and trying to understand how that neuron responds to a stimulus, we can present only stimuli that are evenly spaced in this region that we understand. So what does this border look like in olfaction? What molecules can we smell? So in 1983, Bolins took a stab at this. So this graph, the x-axis is boiling point. So very large molecules are over on the right here. Very small molecules are over on the left. The y-axis here is log P. So this is basically how greasy something is. So things at the top are hydrophobic, very greasy. Things at the bottom are hydrophilic, like to dissolve in water. And this appeared in a review article. There was no text accompanying this figure, how this figure was produced, what data were used to create this. But he just drew this line on here. And he said that anything that falls inside of this parabola has an odor, and anything that falls outside this parabola does not have an odor. So because there's no data, it's hard to replicate this. We had no idea if this was a useful model, how well this worked on a large data set of molecules. but we tried to understand this from some sort of intuitive point of view. So imagine that you're a molecule in a strawberry and you want to be smelled, right? You have to pass a couple checks. First, you're going to have to leave the strawberry. So things that are very heavy, things that are very far to the right over here, are too heavy to leave the strawberry, and you can't smell them. Then you have to sorb into the mucosa at the top of the nasal cavity so that you can actually interact with the receptors that are there surrounded by mucus. So you have to go from this air phase into this liquid phase. So molecules like nitrogen gas, for example, want to stay in gas phase, never sorb at an appreciable concentration, and you can't smell them. Third, you have to get into the receptor binding site itself, the binding pocket. That binding pocket is a little bit hydrophobic. And so molecules that are more on the hydrophobic end of the scale are more likely to be smelled than things that are hydrophilic down here. So that gives us some sense of where this parabola comes, too heavy, too light, or too hydrophilic. But we wanted to see how this would, we wanted to collect a data set and see how this actually worked on a large data set. So we did that. Here I'm showing you the area under the receiver operating characteristic curve. Briefly, for those who don't know, 0.5 means my model is at chance. 1 means my model is perfect. And we took this model from 1983 that was just a parabola and used that to predict if something had an odor. And you can see it has an AUC of about 0.8 or so. We then just threw this into a sort of standard suite of machine learning models to see how well those would work. There's a fragrance-like one that's published. We just use a mini feature model here. And you can see that these models do extremely well at this problem. And it turns out that the reason they do extremely well is that there's very little biology involved here. Most of this is actually a physical transport of getting a molecule to a receptor and has very little to do with biology. So there's a lot less noise than we're used to in dealing with biological organisms. That allowed us to then, from the inspiration of this Bolen's model, go back and try to take this complicated model and simplify it as much as possible. What are the fewest number of features that we could put in here and still perform relatively well? And what we found is that we could get it down to 4 features and still perform almost as well as the model that had many thousand features. And again, this is because it's a very simple transport problem. And so then I asked my postdoc at the time, Emily Mayhew, to try to make an even simpler version so that people could sort of keep this in their brain. How could you look at a chemical structure and get some intuition about whether it would have an odor or not? And so she developed the Mayhew rule of 3. So here, the number of heteroatoms are on the y-axis and the molecular weight on the x-axis. And the rule of 3 is that you want something that, if you want something to have an odor, it needs to be between 30 and 300 Daltons, and it has to have fewer than 3 heteroatoms. And basically 96% of the molecules that fall into this box are odorous. So it's a pretty good rule of thumb for what's going on. So again, not as accurate as these other methods, but gives you some general idea of what's happening here. And then when we have this rule in place, we can go apply these rules to a lot of molecules that have never been synthesized before. So we took a database, the generated database, which is just basically an exercise in generating all the molecules that could be created. And we apply this rule to it. So here, each dot is a particular molecule. The color bar here, the light dots are ones that are likely to have an odor. The dark dots are ones that are likely to not have an odor. And then overlaid in blue here are all the molecules from the flavor fragrance catalogs and publications in the field. And so you can see there are many, many regions of this space where we expect there to be molecules that have a smell, but have not yet been synthesized and not yet been smelled. Okay, so now we have some sort of model that allows us to draw this border around odor space and look at the molecules that have a smell. So let's talk for a minute about variability in odor perception. So as scientists, it's very tempting to think that we're studying universal truths. And so when people come to you and say, well, the thing that I smell is very different from the person that the next to me is smelling, and the sense of smell is very subjective, that is sort of at odds with our view of what we're trying to understand about the world. But we do know that odor and pleasantness varies by culture. So here's fermented herring surströmming, which is very popular in Sweden. It smells like rotting fish. Here's the durian fruit that's popular in Southeast Asia, but smells fecal. Here's fermented soybeans, popular in Japan. And here's fermented walrus from the Bering Strait. All of these are delicacies in these particular cultures, but people outside those cultures find to be very repellent. And so we see that odor pleasantness varies by culture, but at the same time, in the original DREAM challenge, we showed that you can actually predict this pleasantness from chemical structure. So in general, imagine that I give you a set of molecules, molecular structures, and I want you to just organize these so that structures that are very similar to each other are close to each other on a line, and structures that are very different from each other are far apart on that line. In general, when you do that, you'll get a set of structures that look like this, where you end up having things that are small and less complicated on one end, and things that are larger and more complicated on the other end, right? And so this is sort of the first principal component of structure space for odors. And somewhat surprisingly now, if you went down this line and you smelled everything on the left and you kept smelling all the way to the right, what you'd find is that on the left, most of these things smell unpleasant, and on the right, most of these things smell pleasant. And so there's no person involved here. We're just looking at chemical structure. And yet we see this correlation between chemical structure and pleasantness, which is something we think of as subjective. So here's that just with data. So this is from a paper in 2007 from Noam Sobel's group. On the y-axis is the rated pleasantness. Each dot here is a molecule. And then the x-axis here is PC1 of physical chemical space. So basically small to large. And what we see here is that correlation in data, that these two things are correlated in a novel data set. And this group also tested this on 3 different cultures, American, Arab Israeli, and Jewish Israeli, and found that that correlation persisted across these 3 different cultures. So odor pleasantness can be predicted from chemical structure, but it also varies by culture. So how do we keep these things in our head at the same time? Now I want to add one more wrinkle to that, which is that odor pleasantness varies from person to person. So not just culturally, but individuals within a culture will have different perception of an odor. So this is a molecule called androstenone. About 30% of the population can't smell this molecule at all. Another third of the population smells it as unpleasant, smells like urine to them. And then the remaining third of people smell it, and it smells sweet and pleasant like sandalwood to them. Again, to show you that with data, here's the smoky odor guaiacol. So everybody in the room will smell this as smoky, but we'll have different ideas of how intense that smokiness is. And I can look at your genotype for one particular olfactory receptor, OR10G9. And if I look at the polymorphisms there, I can tell quite a bit about how pleasant or unpleasant you're going to find this particular odor. Okay, so all 3 of these things are true. Your perception varies of odor pleasantness varies by culture. It can be predicted from chemical structure, and it varies from person to person. So we set out to do a study to take a look at this more carefully. To do that, we took advantage of a network of field workers who collect new experimental data in multiple cultures that vary in subsistence style, ecology, and geography. So here you can see a wide range of cultures, some of them urban, some of them agrarian, some of them hunter-gatherer, et cetera, from different ecologies. And in this study, we gave them 10 odors. The 10 odors were chosen to span sort of this pleasantness axis that had been collected for the data set that was used for the original DREAM challenge. So we have 10 odors all spaced out on that pleasantness axis according to people in New York City. Each person who was doing the testing was fluent in the target group's native language, so you could communicate with them. And the way that they did this was they actually ranked the odors by manipulating pens that had a particular smell to them. So it'd smell the pens and put them in order from unpleasant to pleasant. And so ranking them avoids these confounds from numeracy and literacy as they do this task. So here's the data. I'm going to show you, this is 10 different odors on my y-axis. And then each column here is an individual person. And the color here is the rank. So I'm showing you on the very left edge here, the mean rank of each odor. So you can see that most people thought vanillin was very pleasant, and most people thought isovaleric acid, which smells sort of like dirty laundry, was very unpleasant. If you look at these sort of gray lines down here, these sort of block out each culture. And what you can see is that it's very difficult to see the lines between specific cultures. Most of this blurs together in a general sense. And across all these cultures, in general, the top is very red and the bottom is very blue. So even though you have individuals who are varying here, in general, you're getting a great description of what's going on just by looking at the average across everybody in the entire study. So let's quantify that. So here we just did it in ANOVA, and this is the eta squared. So it's essentially the percentage of variance that explained by each factor. And so the first thing we can do is just look at the actual odor being used. So which of the 10 odors was being used. And that explains about 40% of the variance, which I find to be a very large amount of the variance in this case. So just knowing what molecule it is tells you a lot about how people are going to rank that. The second is culture. So cultural specific rankings of this explain a really small fraction of the variance, about 7% of the variance. So it is true that culture has an impact, but a relatively small impact on what's going on. And then the remaining category is individual variability. This is actually also includes all the measurement error we have and all the people who didn't understand the task as well is sort of lumped into this category. This is everything that's left over. That's about 53% of the variation. And so now you can sort of understand why we talk about this idea that odor does vary by culture, although that's, I think, exaggerated in these anecdotal examples and is not, at least in this odor set, is relatively rare, that you can make some sort of prediction of what things smell like, and you can also have individual variation in this. So a lot of the times when we run these challenges, we're running panels of people to overcome this individual variation and predict the universal truth here. So regarding this idea that there's certain cultures that have these very strong differences from the rest of the population for specific items, I do want to note that people who smell surströmming, who are Swedish and love this dish, don't find it to smell good, right? They enjoy it in a particular social context when it's presented in the right way. And so there's some argument that the pleasantness here is not as culturally pushed as that anecdote would suggest. Okay, so now I'm going to talk a little bit about predicting the odor of a single molecule. And again, in 2015, we ran a DREAM challenge. The data was collected by Leslie Vosshall and Andreas Keller here. The challenge was organized by Pablo. There are 22 teams that competed in this. And the data set that was used, there are 49 subjects who smelled almost 500 molecules and measured the pleasantness, intensity, and the 19 perceptual attributes of those. So that's shown here. And before this challenge happened, there were models in the field that tried to predict a specific type of odor. So a model that would try to predict if something had a sandalwood odor to it or a musk odor to it. But there were no models that looked at a molecule and generally tried to predict all the features that it would have. And so this really set some benchmarks in the field. The original paper was published in 2017 out of this work. And then you can sort of see this string of papers that came out of this afterward. As the field set a benchmark, people tried to beat that benchmark, as they naturally do. And there's this surge in publication of people trying to make predictions about what molecules smelled like. So it was really transformative for the field. From this original DREAM challenge, the first thing that we noted when we were looking at this was that the predictions were really limited by the data, right? So people were rating a bunch of different features here of this molecule. One of them was how warm does something smell? The other one was how garlic does something smell? And you can imagine that, right here I'm showing you on the x-axis the test-retest correlation. You can imagine if I ask you to describe how warm something smells, that word might be a little fuzzy in your head as to what it actually means for something to smell warm. And indeed, we see that subjects are really inconsistent when they rate this warm attribute. And so then when people built a model and tried to predict how warm something smelled, of course, the model did not do very well. We can't do much better than the test-retest in our sample data set. However, when people are rating garlic, people are very consistent. So the first rating to the second rating, they give basically the same answer. And then when we try to predict that, you can see that the models are much better at predicting garlic for that reason. And here we can show all the data, and you can see in general that test-retest correlation and model test correlation are, of course, very tightly related to each other. And so the conclusion out of this is that we're really data limited in this, both in amount of data and quality of data. We need more and better data to solve this problem, not necessarily better models. So then how do you actually go about raising the bar for data sets in the field? So we took one approach, which was to try to nail down all the features that we cared about and try a bunch of different techniques on a consistent panel. So what criteria were we looking for? So going back to a color analogy, one criteria, of course, things that we care about is resolution. So telling green from orange is not very difficult. You can use a low resolution technique to do that. But if I want to tell these two greens apart from each other, I need a much higher resolution technique. So where on this scale does a particular method fall? And here you can see the RGB codes for this satisfy this. If you're using RGB codes, you can tell the difference between these two dots by looking at these numerical values. Second, we want data that's reproducible. So if I ask you how warm something is 2 different times, I want you to be giving the same answer. So here we're using techniques where you give the same answer both times. And third, we want to make sure that there's efficient data collection. Not only is the quality of the data important, but the amount of data that you get. So which sensory methods best meet these criteria? So first, I want to just give you sort of an overview of different ways that we can use to describe odors. So there's some qualitative profiling methods. One of them is free label, where you just list a bunch of words to describe what something smells like. So it smells like trees, outdoors, grassy, woody, fresh pine. And then another person simply says it smells like roach spray, right? And so you can immediately see some of the problems here. What does roach spray smell like to that individual person? They might use a different brand of roach spray than you do, so there's not a lot of content there. There is some improvement now that we can use large language models to sort of help deal with this type of data, but in the end, people like to use a lot of descriptions that just are very difficult to interpret. Another method is to control the vocabulary people use when they do this. So you're still doing a qualitative odor profile, but you're giving them words like green and floral and fruity and asking them to check which of these actually apply to it. We then moved to quantitative odor profiling methods. So rate all to apply or RADA is very similar to check all to apply or CATA, except now instead of just checking whether it's there or not, you're going to give it a rating from 0 to 5 as to how much that applies to your sample. So this thing is very floral, but just a little bit green, for example. Descriptive analysis is very similar. This is a little bit more rigorous and is usually used with trained panels. They have to answer each of these. They can't leave anything blank for each of these categories, and they use a finer resolution scale. Then there's similarity methods. So one method is to explicitly ask people to rate how similar things are. Using some sort of scale. There's also a method called napping ultra flash profile, which is a hybrid method. Basically, you're asking people to do principal component analysis on a data set. You give them a set of 10 jars and then have them space them out in a 2D space on a mat in front of them and put things that smell similar close together and things that smell different far apart, and then write down some words about why they cluster them in a specific way. So a bunch of these different methods are out in the field, and we wanted to use a consistent panel. We used actually 2 different panels. One of them was a lightly trained panel. They did about 4 hours of training with us in the lab before they started. And one was a highly trained panel. They've been doing this for 23 years as part of an industry panel, right? The minimum number of years of the people on the panel was 23 years of experience, smelling products, doing descriptive analysis on those products. Basically what we found is that, first of all, similarity methods don't scale very well. So they scale with a square of the number of samples you have to measure all the pairwise similarities. Even if you do some tricks to reduce that, they still don't scale particularly well. RADA scales, RADA-CATA, a napping scale much better. We looked at the test-retest correlation. We found that they're relatively comparable across these methods, but the resolution of RADA was much higher than that of CATA or the snapping ultra-flash profile method. So for a lot of the data that we collected, we started using RADA. So that said, while we were busy trying to improve the quality of the data we were collecting, our collaborators at Google Research took a slightly different approach, which is that they bought 2 giant data sets so that they could use them. And so this data is actually, I would say, much lower quality in the sense that this is what it looks like. You get 1 molecule, and then you get a description like camphoraceous, cooling, minty, right? So you have 3 words. You don't know if one of those applies more than the other 2. For the odor, the words that don't appear here, we don't really have a good idea of if this does not smell fishy, or if people just left that off because it was a very minor note, right? And so despite the fact that this was a sort of a lower quality data set, there were 5,000 molecules, almost 5,000 molecules in this data set. So the green squares here represent 100 different molecules from Good Sense, the blue from Leffingwell, and in orange are molecules that are in both data sets. So having this really large data set, the original DREAM challenge was based on 500 molecules. Now we have 10 times that amount of data, allowed us to make a much better model. So when people try to map this odor space and look at these different molecules and how they're related to each other, they note a couple different things. So if we take this data and we do principal component analysis on the true labels, there are a bunch of gray dots here, and each gray dot represents a molecule, one of the 5,000 molecules. And then we've put some contours around specific percepts. So you can see the sort of floral, meaty, and ethereal. And you can see that the floral and meaty categories don't overlap. So this is something that if you smell a lot of molecules, you notice very quickly. Things that smell floral don't also smell meaty. And then within that, you also see these sort of hierarchical organization. So you have 3 different types of floral smells that all fall within this overarching contour. You have savory, beefy, and roasted, which all fall within this meaty contour. So you have both this separation at the high level, and you have this hierarchical organization within. So this is the way that odors are organized perceptually. Now let's take this map and take exactly the same molecules and make a map that's based not on perception, but based on chemical structure. So we're just taking a chemical and looking at how similar it is. We're going to space out all these gray dots on this again, and then we overlay contours. And what you can see is we've taken this nicely organized map where you have these different qualities spread out, and you've essentially crumpled that map up now. And so all these different percepts are touching each other. So a lot of the work that we're doing in the machine learning here is trying to uncrumple this map, recover from chemical structure where these things are. You can see that it's not a clean correlation where things that are similar in chemical structure are also similar in terms of perception. And so the way that we set out to uncrumple this map is to use a neural network. We use a graph neural network. So a standard neural network is going to feed in different molecular features here and have some intermediate layers and then some prediction layer that predicts how citrus it is, how baked it is, et cetera. In the graph neural network, we simply are changing the type of data that we feed in at the front end. So you represent each molecule as a graph. You have features for each node, like atomic number and formal charge. You have features for each bond, like degree and aromaticity. And those are allowed to interact with each other as they feed into the network. So you can look at sort of local structure in this molecule and have that be a feature for making predictions. So this last layer will tell you how citrus something is, but the next to last layer is actually really interesting. This is the embeddings that sort of map out this odor space. So we can actually look at that penultimate layer, pull that out, and map things using that. So just to remind you of the maps, this is a perceptual map. This is a raw chemical structure map. Now I'm going to show you the map from the embeddings, which we call the principal odor map. And what you can see is that even though we're deriving directly from chemical structure, we're able to recover a lot of this perceptual structure. So again, floral things and meaty things don't overlap. And in general, the different types of flowers fall back into this hierarchical arrangement. Okay. So although you can now use this to sort of visualize how it's able to make good predictions about things, let me sort of quantify its performance. So the first thing that we did was do a prospective validation. So the model made predictions for a set of molecules, and then we collected human data on those molecules to see how well those predictions worked. To get the molecules that we wanted to predict, we set up a pipeline here. We started with an e-molecules catalog. This is a chemical library primarily designed for drug discovery, so generating lots and lots of different chemical structures that you can purchase, about 7 million molecules that we could have bought. We then set up some filters. So first we wanted to filter for things that actually have an odor. So most drug-like compounds are too heavy to be volatile, for example. We wanted to screen all of those out. We wanted to keep our price reasonable, so we set a price filter. And we wanted them to be non-toxic so that we could actually deliver them to human subjects and have them smell them. So we had about 89,000 molecules left at the end of that. And then from that set, we picked 400 to test the external and the prospective validation. The way that we chose from that last 89,000 down to 400 was we set up a couple rules. So the first one is that molecules have to be structurally distinct from each other. We don't want the model to be cheating by basically picking a bunch of molecules that are all very similar to each other and solving it that way. We wanted them to be spread out in perceptual space, so we want to make sure that the predicted labels cover the whole gamut. And then we wanted to make sure that all the molecules that we picked were structurally or perceptually distinct from any of the training examples that we used as much as possible. So we wanted to make this challenge hard for us to solve, in other words. The way that we collected the data was we did this rate all that apply method. So the panel was trained in how to do that. So rather than just asking people to rate how green something is, we actually give them a jar that smells green. So it has a molecule or a substance in it. So they can use that as a reference point. So when we say, how green does it smell, they know exactly what we mean with a universal reference. They are given a 55-word lexicon to use for this. So they rate how all those 55 words apply to each molecule. Here I'm going to show you the data for one particular molecule, shown at the top here. So at the bottom are the y-axis here are our panelists, and the x-axis are the 55 different odor descriptors. If it's a dark green, that means that they rated it as a 5. That particular label is very applicable to this odor, down to a 0, which means it doesn't apply. And you can see here, as I showed you before, there's a lot of individual variation in our panelists. You can see that each row here is slightly different from the next. However, if you average across about 15 to 20 people, you start to get a very stable representation. So if I get 15 to 20 people to describe something and another 15 to 20 people to describe it, you see that those are very tightly correlated with each other. So now this is now our ground truth. So we basically are averaging these subjects together. The top 5 labels here, in this case, are sweet, fruity, vanilla, powdery, and floral. So that's what that smells like. So we have no ground truth except for what the human panel gives us. We then go and unblind the model predictions for this. So I'm not showing you the best molecule here. I'm showing you something around the 80th percentile of performance in the model. And you can see that in this case, it got 4 of the 5 labels correct. And then it also predicts this label, which no human panelists ever picked. It describes this molecule as sweet, vanilla, powdery, floral, and hay. So hay is the descriptor that's extra, and it missed that this smelled fruity to the panel. Now, to quantify this, we are going to take the panel average here as our ground truth. And you can see that the best subject here correlated with the panel average with a R value of 0.52. The median panelist had a correlation of 0.18, and the graph neural network had a correlation value of 0.63. So just to put that in context, here on the y-axis, I'm going to show you basically the difference in median correlation relative to the human baseline. So this is the median panelist performance is shown as this 0 point that we're normalized to. I'm going to show you a shuffled model here, and you can see the performance is much worse than the median panelist. I'm going to do 2 k-nearest neighbor models here using either fingerprints or physical chemical descriptors. I'm going to show you a random forest model. This is the model that won the previous DREAM challenge applied to this. And then I'm going to show you the graph neural network. And surprisingly, this actually beats the median panelist. So we now have a model that is able to replace the average person on a sensory panel and improve the overall panel average. So I showed you that the model surpasses human performance in aggregate. I'm just going to break that down a little bit. How does it work across perceptual classes? So here are all the different descriptors, the 55 different descriptors. Some of them, and this shows you the correlation to panel mean. So some at the top here are ones we perform very well with, like garlic, fishy, fruity, meaty, and camphor. Some of the ones at the bottom here that we're worst at, musk, hay, ozone, and fermented. Then you can also see, and so I'm showing you, sorry, here the gray dots are the panelists, the orange dot is the graph neural network, and the purple diamond is the random forest, which is very similar to the model that won the DREAM challenge, the original DREAM challenge, as a baseline. You can also see that the amount of data that we have is really important here. So here I'm showing you the amount of data on a log scale, the amount of training data we had for a particular descriptor. So there are lots of molecules here that smell fruity, but there are relatively few that smell like musk. And then I'm showing you the correlation with panel mean. So high performance at the top, low performance at the bottom. And you can see there's roughly a floor here, that once we get a lot of data about what something smells like, we're able to make pretty accurate predictions about that. And so again, we think that the amount of data we have is a real limiting factor here. Okay, so I showed you that the model outperforms current benchmarks, which is really important. But what's more exciting is that this model actually has learned something about olfaction, learned something about odor space beyond exactly what we taught it. So what we did was apply it to a bunch of different other data sets to see how well it translates to those. Some of those data sets are very similar to the data set that we collected. So Dravniak's Atlas and Keller are both basically using the same method where you give people a word and they see how well it applies to something. We do 3 different models here. One is we just use sort of that crumple map I showed you. We just take the chemical information and do a linear model on that. Here's same chemical information doing a support vector machine, all of these untuned. And then I'm doing principal odor map. Again, this is just a linear model on top of the principal odor map coordinates. So all we're doing is feeding in the coordinates for something and building a simple linear model. And that immediately outperforms these other 2 based on that crumpled map. Perhaps not surprising because the data set's relatively similar. Here I'm doing the same thing with the data from the DREAM challenge. Again, immediately outperforms simple models based on that crumpled map. Here I'm showing you a slightly different data set. Rather than saying how green does something smell, this just measures similarity between lots of pairs of molecules. And again, principal odor map is doing well at that. The 4th one is the most surprising. So this is a data set of detection thresholds. So this is the concentration, the minimum concentration people can detect. And so we have never ever taught our model anything about intensity in any explicit way or about detection threshold. And yet when we apply it to this, it also outperforms these other two models. So the way that we think about this model is that it's representing each molecule in some sort of vector. We can project that vector down to the two different axes. So for example, green or fruity, and know how green or fruity this smells. We can measure the distance between two different vectors in this space, and that gives us how similar they are, which allows us to predict this SNITS model. And then interestingly, the length of this vector also seems to predict the detectability of a molecule as well. So even though we've never taught it about intensity, it has still somehow learned this information. So then some collaborators on this project also went on to do another paper where they extended this even further. So rather than just look at olfactory data sets, they pull, in humans, they pulled data sets not only from humans, but also from animals. And then they also pulled some other non-olfactory data sets. So up here you can see some data sets from mosquitoes or rats or fruit flies. You can also see some things like electronic properties or binding affinity to a GPCR or hydration-free energies, sort of more physical properties of a molecule. And what they found is that when they basically used the principal odor map coordinates versus some general chemical features, and whenever you're looking at an olfactory data set, you see that these dots fall above the line, indicating that a principal odor map is better than a general chemical feature map. And if you look at these sort of orange dots, they tend to fall below the line, meaning that this isn't a magical thing that solves everything. We don't know anything about general GPCR binding. We instead know something about olfaction specifically. So this principal odor map, this is basically a set of 500,000 molecules that could have a smell and could be synthesized. The RGB values at these are the first three principal components of perceptual space. And so if this were a perfect map, you would never see any purple in the middle of these red zones here. So it's not a perfect map, but you can see relatively smooth contours between these areas. This map faithfully represents the perceptual hierarchies and distances that we see in the training data. This can be extended to novel odorants, and it generalizes to other olfactory tasks. So I want to note this is not a perfect system. So one thing to note, which I think is pretty entertaining, is that we asked a panel to describe these odors, and here's one example. They describe this as the top 4 descriptors: sharp, this is 1.8 out of 5, sweet, roasted, and buttery. And so this is the average across our panel of 15 people. We gave the same molecule to a master perfumer, Christophe Lodemiel, to smell, and asked him to describe what it smelled like. And he described it as ski lodge, fireplace without a fire. Right? So you can see that our description is missing some of the poetry here that we get from our master perfumer. Here's another example: musty, ozone, earthy, and medicinal. And when he smelled it, he described it as the hot tub is near. So you can see that we're getting some description of the elemental features here, but we don't really understand what this odor is. Okay, so in addition, this model is really only looking at single molecules. So we have no way to talk about mixtures. And unfortunately, pretty much every odor that we encounter in daily life is a complicated mixture. So here is a GCMS trace. The x-axis here is basically a time axis, which just shows you the volatility of a molecule. So you're separating out molecules by how fast they pass through a column and how volatile they are. And then on the y-axis tells you the abundance, so how much of each molecule there is. So each peak here is a different molecule that's coming off of the GCMS. And here I'm just going to show you some of these. This is actually the smell of coffee, so we put coffee through here. All these are molecules that are in coffee and actually contribute to the flavor of coffee. So you can see that mixtures are way more complicated than the things that we're using in lab. We're giving single molecules, and you have these very complicated mixtures. However, I do want to stress that when you first look at these GCMS traces, you might be overwhelmed and think this problem is way, way harder than what we've been doing. But mixtures are not really as complicated as this GC trace initially suggests. So first of all, many of the molecules that you see there, even though the peak is very high, may be something that's very difficult to smell and is actually present at a level below your detection threshold. Food scientists have been studying these complex mixtures for a long time with the idea of looking at this GCMS trace, picking out which molecules are essential to reproduce that, and then creating a flavor to match it. What they see is that in general, only about 10% of the volatiles that they see in a GCMS trace are actually needed to reproduce this. So here's a review paper. On the y-axis here are what they call key food odorants. These are odorants that are necessary to reconstruct a particular odor. And then on the x-axis here are food samples. So they're pulling from a variety of different categories of food. And then you can see this graph of abundance. So you can see that this molecule here is a key food odor that's present in almost 50% of the foods that they tested is necessary for reconstitution of that. Molecule is methanol. It smells like roasted potatoes. And what they see is that you only really need about 270 molecules to reproduce all these almost 300 foods. And so this has not been rigorously tested. It's not clear these make exact replicas of these foods, but the point still stands that you need relatively few molecules to recreate lots and lots of different food odors. Also encouraging, there are already 2 models that make some sort of predictions on mixtures. The first one is from Noam Sobel's lab in 2013, which then was later updated in 2020. This is basically a relatively simple model. They represent each molecule as a 21-dimensional vector. When you mix them together, you basically are averaging those together. And when you want to compare 2 different mixtures to see how different they are, you just measure the angle between those vectors. Here is a graph with the angle distance on the x-axis and the normalized similarity rating from human subjects on the y-axis. And you can see that it performs relatively well. The 2020 model updated that. In the original version there, all these molecules were balanced for intensity, so they all had the same intensity, which is of course not anywhere near what we see in nature. In 2020, they added a filter for intensity as well as doing some other things. Here's another paper that does that from Pablo's group. So here as a baseline, these are the 4 data sets that were in the DREAM mixture challenge that was just run. And here are sort of the baseline RMSEs for these. When you make a direct prediction from chemical structure to percept, Pablo's group did an intermediate step where they first took chemical structure and mapped it to semantic descriptions. So they used the original DREAM model to say how woody is it, how green is it, and then use that to make final predictions. And you can see that RMSE drops, indicating the model performs better on all 4 data sets when you add that intermediate step of understanding perception. Okay, so both of those models suggest that mixtures aren't impossible to predict, but I want to know that there's a really persistent idea in the field that the olfactory code is not nearly as simple as, for example, the code for color vision. And so what do I mean? So here are 3 odors, X, Y, and Z, and here are a bunch of receptors. A blue dot means that this odor activates this receptor. And so you can see the different patterns here of activation. They then take a mixture of X plus Y, and under one theory, this model 1 of non-modulation, if you mix X and Y together, you activate all the receptors that were activated by X or by Y. So you're basically, anything that was activated by either component is now active. And so as you get larger and larger mixtures, you can be activating more and more receptors. And we definitely do not see this in the olfactory system. If you give a mouse a monomolecular odor and look at how many glomeruli light up, and then you give a mouse a complex odor from their nest that has urine and feces and hay in it, it activates about the same number of glomeruli. So we don't see this huge expansion in number of responding cells. And so how do we explain that? We know that there are a number of different interactions among receptors. So for example, even though X activates receptor 2, maybe Y is an antagonist and so blocks that receptor site. And so when you have X and Y present in the same mixture, that receptor is no longer activated. You can also have cases where even though neither odor is activating this receptor, when they're in combination, they do activate it. So we know that these interact this happens at the receptor level. And so because of all these nonlinear interactions, a lot of people believe that you're going to need to build a model that understands all these pairwise interactions. So just understanding what receptors are activated by each single molecule is not going to be enough. And so this is somewhat at odds with this idea that we're using these relatively simple linear models to make predictions, and they've been relatively successful. And so how much this is governed by this situation versus linear models is still unclear. So just to wrap up, we can predict if a given molecular structure has an odor, and we are quite successful at this, primarily, I think, because it's mostly a physical process rather than a biological one. We can predict odor valence from molecular structure across a bunch of different cultures and subsistence styles. And we can predict odor character from molecular structure and outperform a median panelist at that task. And so we're very excited to go forward and understand mixtures now, and hopefully we'll learn all about that in the next session. Finally, I just want to thank a bunch of people who in my lab who contributed to this, close collaborators, Rick Gherkin, who was at ASU, a group at Google Research who contributed the principal odor map model, Jane Parker, who did a lot of quality control on that, the various funding agencies, and then both Rick and a number of people from Google Research formed a startup called Osmo that's now taking this forward and trying to pursue a commercial path with it. And I'm happy to take some questions.
If anybody wants to ask a question, I think it's better if you come to the microphone so that the audience can hear, if you don't mind. Hi, Benjamin.
Thanks, Benjamin Epkins.
Other keynote.
We need to help each other, right? Very interesting talk. I was wondering the, what do you call it, the perfume industry.
Yeah.
They must have tons of data. I don't know what kind of data, but is there any way you could leverage those and get access to those data?
Yeah, so there is a lot of data in the perfume industry. Unfortunately, it's not very standardized. So we don't know about everything that's out there, of course, right? But from the people we've talked to, there's very little in the sense of large standardized data sets that span sort of a wide range of chemical structures. So for example, a lot of times people run a very focused study. They're trying to develop a better coffee, and they spend a lot of time understanding coffee. They develop a specific vocabulary for coffee, and they have a panel describe lots and lots of samples. But those samples are very narrow in terms of perceptual gamut that they're spanning, and they use very specific vocabulary to do that. So then stitching all that together is very difficult. The Leffingwell and GoodSense data sets were actually two of the larger data sets. So a lot of the flavor fragrance companies were purchasing Leffingwell and using that internally as sort of a reference. And GoodSense was sort of a well-known website resource in the industry. It's still a little unclear exactly how that evolved or where that data, either of those data sets exactly come from, but they were used widely in the industry. So I think there is some data out there. I think there are people who are trying to unlock some of that data. We've tried to unlock some of that data, but it's very difficult. There's a lot of secrecy in that industry, and they protect things pretty carefully.
And do you know if industry is using data science and machine learning and whatnot internally?
Yeah, absolutely. So I don't know exactly when all these groups were formed, but most of the large flavor fragrance houses now have an internal data science team. So it has come online. It's not clear exactly what they're doing. So in some cases, it seems clear that a team that was originally looking at this type of question was then switched over to look at, for example, skin toxicity. So that became a very important thing in the company. They wanted to make sure these ingredients were not causing allergic reactions. And so they basically dumped everybody on that team to look at that problem. So again, this has some commercial value to it, but it's difficult to spend a lot of resources in a very general, wide-ranging model on this topic. Yeah.
Sorry, is this good?
Yeah, but
We have to touch. Thank you, fantastic talk, and it's very nice to see how the first DREAM challenge had an influence in the benchmarking of these things, though there is a lot of work to be done.
Yeah.
My question is about, besides the verbal description, have there been studies of, this might be very difficult to collect this data, but fMRI or EEG to understand whether there is something that goes beyond what we can verbalize?
Yeah, so there are some data sets. In general, they tend to be relatively small, and that's sort of true of olfactory data sets in general. And part of the reason is that for visual data sets, you can collect a lot of data very rapidly. And in olfactory data sets, you have an adaptation period. So basically, when I smell an odor, we usually wait for about 30 seconds before we present another odor. And in fact, you should probably wait even longer than that to totally get rid of the influence of the first odor. And so, whereas in vision, you can show a stimulus every couple of seconds, we're delayed by about 30 seconds in olfaction. And so collecting data, large data sets is very difficult in general. And then if you add on top of that the number of averages you'll need from EEG or fMRI, those data sets are relatively rare. So there was a recent publication from Torsten Kahn's group where they did I don't know, I think about 100 odors in fMRI. So collecting a huge amount of data on that. And they were basically scanning people for, I forget, hundreds of hours, I think, of scanning on people, so over different sessions, to try to collect enough data to do that. So it is possible, but it's very rare.
And was there any interesting results as far as understanding something that you don't understand with these methods?
I don't want to say no, but I can't think of any like major thing that stands out from it, but I'd have to reread the paper.
We have a question online. You mentioned that pleasantness is culture specific. I'm thinking about tests of olfactory ability like UPSIT. Should these tests also be culture specific?
Yeah, so the answer is yes. So there has been actually a bunch of data on the UPSIT. This is a standard 40-item scratch and sniff test they use for clinical diagnosis. So you scratch off an odor and you have 4 choices. You know, does it smell like grass or oil or water or cucumber? And you have to pick one it most smells like. Obviously, that has a lot of cultural bias to it in terms of what words you're presenting, what language you're presenting those words in, what specific odor stimuli you're using. Those have actually been normed across a bunch of different cultures, and they have made cultural specific versions of those tests in specific cases. So yeah, it is important if you're doing some sort of clinical diagnosis like that, that you address that problem.
Hi, Dr. Mainland. I really enjoyed your presentation. It was quite fascinating. I want to go back to your pleasantness versus the size of these chemical structures. I thought that was super interesting. My question is, did you guys look at particular chemical species or chemical structures, like in an organic chemistry sense, like trying to see if, like, I don't know, branch structures or different molecules had certain pleasantness? I'm surprised I didn't see a follow-up on that.
Yeah, so there are a number of different ways that you can predict pleasantness. And so, I mean, a lot of people know that sulfur is unpleasant, right? So a sulfur molecule tends to smell like rotten eggs no matter what else is happening in the structure. An ester tends to be fruity. So there are a lot of these correlations that you see, and a lot of the sort of machine learning is basically picking those things out and learning how to do that. So some sort of very basic linear models actually did quite well in the original DREAM challenge that were just using these type of physical chemical features. So it's definitely possible to do. But I mean, I think it's still striking that the size was such a thing, that was such a big factor there. I think I have no real explanation, but I have a hand-wavy explanation, which is that if you are an organism and you have a lot of resources, you're resource rich, you have enough, you can create large molecules. And if you are resource poor, you cannot. And indeed, if you actually start decaying, you tend to be decaying due to entropy down into smaller molecules. So not every molecule can also spend a lot of resources to make a very, terrible toxin. And so it's not always going to be pleasant. But if you just look at the average over everything, in general, you'd expect small things to be more unpleasant than large things.
Thank you.
Yeah.
Joel, I have also a question about if you could comment a little bit. You did some studies on how much the genetics tells you about, how much predictability there is in genetics. Can you just comment on that?
Sure. Yeah. So we did a number of studies where we, we had about 400 people smelling a bunch of odors, and primarily rating intensity and pleasantness of those data sets rather than actual odor quality, just because it's very fast to get them to quickly rate those. And then we would do a genome-wide association, or in some case, exome sequencing, and look at how well we can predict that. And we see a huge range. There's some molecules where, so beta-ionone is a very famous one, where a huge percentage of the variance is explained by basically one polymorphism. So this is a molecule that smells sort of floral and candy-ish. And if I know your genetics for that, I know almost perfectly how intense you'll find it and what you'll smell it as. And then a number of things we have very little explanatory power for. So there's a huge range in how much we can explain from genetics.
We have one more question online. The GNN only looks at atoms and bonds, but doesn't know about 3D structure. Is there scope for methods that use 3D information, such as binding to ORs or the like, into account?
Yeah, so absolutely, there are ways to improve on these. I think one of the huge developments in the field is that the first structure of a mammalian OR was discovered, a year and a half ago now. And so, and AlphaFold is starting to make predictions about binding, although it's not quite perfect for olfaction yet, but clearly the field is heading in that direction where we start to understand how these receptors are activated and can add that source of information to that. In theory, that's of course going to improve what's going on, and we've already seen actually, we'll hear one talk from this particular DREAM challenge that used some of that information about receptor activation. So yes, there's more structural information coming out, there's more understanding of what receptors are activated, and all that should improve the models.
Finally, just a more general question. Have you ever thought of how much that we learn in olfaction can be applied to just drug discovery in the sense of, pure molecules, GPCRs, mixtures of molecules? I mean, it just seems to be really two parallel things.
Yeah, correct. Yeah, so the Google research team was actually, the olfaction team was actually embedded with a bunch of people doing this type of work on GPCRs in general. And of course, almost half of the current drugs on the market target some version of GPCR, so it's a huge thing to go after. A lot of these things relate, and actually what's really nice about the olfactory system is that we can sort of rapidly test these things. So giving people lots and lots of drugs and seeing how they respond to them is not a practical thing to do, but giving them lots and lots of odors and having them respond to that is a practical thing to do. And so those teams were closely aligned, looking at each other, what each other were doing to try to understand how they could take these learnings from olfaction and apply them to drug discovery. Yeah.